and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Richard Johnson, thank you very much for joining me.
It's a pleasure to be on your show, Nick.
Do you want to start off by giving everyone a little bit about your background, who you are, and what you've studied in your career?
Sure. So my name's Richard Johnson, and I'm a professor of medicine. I did both clinical practice as well as scientific research. And I've been involved in studies of sugar metabolism since the late 1990s. I've been funded by the National Institute of Health and other organizations. I'm highly cited, published hundreds of papers, and I have a strong interest in fructose, and especially how it might have a role in disease today.
Fructose, one of the major simple sugars that's out there. You've got glucose, fructose, often found together as sucrose, table sugar. Obviously, we've got high fructose corn syrup. We've got whole fruits. We've got processed foods. Lots of different places that fructose shows up in the modern human diet. I just want to talk a little bit about glucose versus fructose metabolism, especially in the liver, and give people a basic sense for how these things are used by the body, broken down, and so forth. So can you give us, in pretty basic terms and plain language, the major differences between glucose and fructose metabolism in the liver? How are our bodies actually utilizing these two simple sugars differently?
Yeah. Well, let's just begin with the very basics there. Glucose and fructose are two simple sugars, 6-carbon sugars. In many respects, glucose is the fuel that we're most used to talking about. Glucose is the principal carbohydrate that is utilized in mammals and humans. And we have high levels of circulating glucose. It gets stored as glycogen. And it is used as our main carbohydrate fuel. Fructose circulates in our blood as well, but it's really about almost 1/100th the concentration of glucose. So it's circulating at a very low concentration, but it turns out to have an extremely important role in human metabolism as well. Fructose, the main dietary sources are honey and fruit, the main natural sources. And many people think of fruit and honey as a very healthy types of food. But fructose is also in table sugar. And it turns out that table sugar is a dimer of glucose and fructose. They're bound together. And high fructose corn syrup is a mixture of fructose and glucose, free sugars kind of mixed together. Now, the reason we like sugar so much, and people have a sweet taste for sugar, we have a taste bud that just for sweet things, and it gives us a pleasure response when we eat this stuff. And it actually is for both glucose and fructose. Both of them activate that sweet receptor. But fructose is viewed as a sweeter sugar. And it turns out that extra sweetness is what makes sucrose or table sugar so delicious. And high fructose corn syrup was built on that. When they made this, they combined free glucose and fructose, and they found that people actually prefer a little more fructose than glucose in taste testing.
Yeah, yeah,
And so high fructose corn syrup, at least in soft drinks, is typically 55% fructose and 45% glucose.
yeah. So
So the two
glucose,
sugars are fairly similar.
yeah,
So now when you ingest fructose, it has its own transporters in the intestine that are separate from glucose. And those transporters are important in getting the fructose into the intestinal wall where it's maybe a fairly significant amount gets metabolized. And then the rest of it gets into the liver. And in the liver, again, there's a very, what we call a first pass effect, where a lot of the fructose is metabolized in the liver before it ever sees the circulation. So only about 10% of the fructose we ingest will typically get past the intestinal and liver metabolism. So the liver preferentially loves fructose. And when the fructose escapes the liver, some of it will be passed into the urine, where it will be taken up in the kidney, in the tubules, because they have that transporter. Some is taken up in fat cells, some is taken up in the brain, and then other places like the islets of the pancreas. Now, when fructose is metabolized in the liver, it is metabolized preferentially by a different enzyme than the one that metabolizes glucose. And so there's a whole different enzymatic pathway. That enzyme is called fructokinase, and it's an enzyme that I have heavily studied for decades. And that enzyme loves to metabolize fructose pretty much in an unregulated way. In other words, it will metabolize it as rapidly as it can with no feedback. Now that allows, when fructose and glucose get metabolized, the early beginning of metabolism involves a phosphorylation of fructose or a phosphorylation of glucose. So glucose gets phosphorylated at the sixth position and then gets broken down eventually to generate like pyruvate, and lactate in the glycolytic pathway. But also then it can feed into the Krebs cycle and the electron transport chain to generate a lot of energy. Fructose theoretically can do the same. But what happens is when fructose gets metabolized, that initial phosphorylation by ATP is so rapid that ATP levels fall, and there's no feedback system to stop that. And when the ATP levels fall, intracellular phosphate also falls.
mm-hmm.
That activates a whole different type of enzyme reactions that lead to different consequences
Yeah,
compared to
yeah.
glucose.
I think this is a great place to pause because we've already covered quite a bit. And we're going to
Okay.
circle back to this, especially the lack of feedback and why there's a lack of feedback and how that ties into maybe evolution stuff. But let me just kind of like compress and repeat some of what you said for people. And
That's
let's
perfect.
start in the tongue and work our way down. Let's just pretend we just eat a tablespoon of sucrose. So you've got glucose and fructose in there. They're both sugars. They're both sweet. We like both of them. We have a pleasure response to both of them. Fructose is actually a little bit sweeter. And the reason that something like high fructose corn syrup is 55 fructose, 45 glucose is actually because food companies did experiments, they did trial and error, and they basically locked in on that as the ratio that people sort of like the best. It's the most pleasurable
That's right.
sweetness ratio. And so right on the tongue, there's a little bit of a difference between fructose and glucose. We swallow it. And then once we get into the gut, there's some big differences. Fructose isn't going to make it into circulation, as you said, as much as glucose is. It's going to get metabolized by some cells in the small intestine. It's then going to basically go to the liver and mostly get metabolized there. It's not going to go into general circulation nearly as much as glucose.
Yeah, so most dietary fructose, and I mentioned dietary because our bodies can also make fructose. So dietary fructose is pretty much only a small fraction of it gets into the circulation.
Mm-hmm.
However, dietary fructose appears to activate endogenous fructose at different sites in the body. So if you drink a soft drink, which has a lot of fructose in the diet, very little of that fructose makes it to the brain, but the brain will start making fructose in response to the soft drink.
Got it. So there's fructose in the diet that we put into our mouths and swallow. There's also fructose that can be produced internally in response to that exogenous fructose. And we'll get into some of that. But most, if you eat an apple or you eat some honey or whatever, you're going to swallow it. Most of that fructose will be metabolized by some cells in the intestine and then go to the liver. Very little of it will enter circulation and go elsewhere. Now, if we just think about this at a very high level, in very basic terms for people, normally we think of the liver as like a detox organ, and we think of the kidney as a filtration system. Why would it be that fructose is going to be mostly handled by the liver and not sent to all the other cells like glucose is? Like, what does that start to tell us?
Okay. So it turns out that the liver doesn't just remove toxins. The liver is the central site that drives the metabolic syndrome, including obesity and so forth. So if we, for example, if we knock out in a mouse, we knock out the fructokinase just in the liver, the animals will be fully protected from sugar-induced metabolic syndrome. And that includes obesity, insulin resistance, fatty liver, all those kinds of things are protected if you knock out fructose metabolism in the liver. So it turns out that the liver actually has a very central role in controlling metabolic features. Now, interestingly, it does it through a communication with the brain. So there's a very strong liver-brain connection that is involved in this, as well as the liver-fat connection. And one could argue that the brain also has a major role, but when you knock out the liver, you affect the brain signaling. And so it actually blocks some of the changes that occur in the brain. So the liver is really central. Now, what's really interesting is that if you knock out, I'm just going to tell you this because it's really interesting. If you give a sweet food, even like artificial sugar, like Splenda, sucralose, it will also activate the sweet receptors in the tongue. But if you knock out the sweet receptors, so there's no sweet receptors, mice still like fructose.
So it's not just
still like glucose, but they do not like artificial sugars. So the artificial sugars are working strictly through the taste receptors in the tongue, whereas the fructose, it turns out that if you knock out the taste sweet receptors, animals will eat a little bit less fructose and they'll eat less glucose. So there is a direct tongue taste bud mechanism driving increased intake, but the animals will still still want fructose, and they'll still want glucose.
Got it. So when you consume glucose and fructose, the tongue is perhaps the first time information can go to your brain about what's going in the body.
That's right.
But there's also a parallel pathway going from the liver to the brain. So even if you didn't have a tongue or you had some kind of weird mutation where you had no sweet receptor on your tongue, the brain would still be informed about that sugar via the liver.
Right. So if you knock out, this is really interesting. If you knock out the fructose enzyme in the whole animal, they don't like sugar anymore, or they don't like fructose anymore. I mean, they like it, they'll eat it, but there's no desire to eat fructose. So the fructose effects are really centered not on the sweetness, but on the metabolism. But here's what's really interesting. If you knock out fructose metabolism in the intestine, the animals don't like fructose anymore,
Hmm.
but they still get fat with fructose. If you knock out fructose metabolism in the liver, they love sugar, but they won't get fat
Hmm.
and they won't develop metabolic syndrome.
So one of the implications here is for metabolic syndrome and those types of things, something has to happen in the liver that's critical
Right.
here.
That's right. And it turns out that addiction and craving can be separated from the metabolic effects. You can crave sugar like a liver knockout, craves sugar, it'll eat as much sugar as it can, but it won't get fat. So you can separate craving from metabolic syndrome.
Right. And that's interesting.
But
And
they're
it's also,
both involving fructose.
yeah. Right. So that's interesting. And so, something has to happen in the liver. The liver is sensing the fructose, and then
Correct.
metabolic effects that we're going to unpack happen. And that's going to end up being critical for some of the bad things that happen downstream of fructose ingestion at high levels. Let's start to talk a little bit about what's actually going on in the liver in response to fructose ingestion. There's a number of things I want to talk about here. And so I'll lay those out and you can kind of take us through the sequence you think is the best to help people understand. But some of the things that happen are fat synthesis in the liver called de novo lipogenesis, oxidative stress, reactive oxygen species related biology, and then ATP depletion and uric acid. So between those things and anything else you might want to touch on in the liver, what's happening when we eat fructose inside the liver, and why is this stuff so consequential for health?
Okay, so we're going to go into biochemistry here. And I should mention that we just had a review in Nature Metabolism that really lays out the metabolite, the biochemistry for those who want to read about it. So it turns out that when you eat fructose and it gets to the liver, that this initial phosphorylation of fructose leads to a drop in ATP and a drop in intracellular phosphate.
So the liver is using cellular energy to process the fructose right off the
That's
bat.
right. So there's a sudden drop of energy in the liver, which you can measure in animals and humans, and it's been shown to occur with even one soft drink.
Hmm.
It's a rapid burn and then a crash, which is clinically seen by people. A lot of people seem to think that their children will show this, where there's a period of hyperactivity and then a crash. And it sort of times with the rapid consumption of fructose and ATP depletion, burning, and then a period of time when the ATP levels are low. The ATP levels, when they drop, initially it's from consumption, but when the phosphate falls, it activates an enzyme called AMP deaminase, which sucks off, when ATP is consumed, it generates AMP. And normally AMP is recovered back to ATP. But this enzyme sweeps the AMP away to make uric acid. So ATP levels can't recover very quickly because the substrate to make ATP is AMP, and that is removed by the activation of this enzyme that's specific to fructose. And that enzyme generates uric acid. Now, uric acid is a breakdown product of ATP, but it's also a breakdown product of RNA and DNA. And we all have uric acid in our blood, and it varies quite a bit in people. But when you eat fructose, there's a rapid generation of uric acid, and it will increase inside the cell in the liver, but it will also spill out into the blood.
Hmm.
And so the uric acid levels go up in our blood about 15 minutes after we eat a high sugary food.
Got it. So one can of soda, that's roughly the amount of fructose that can do the things we're talking about. If I
Right.
drink one can of soda that's got, tens of grams of sugar in it, let's say, two signals in the liver right off the bat, they're going to happen pretty quickly. One, the ATP drops in the liver. So the liver is going to sort of detect that as running lower on the energy currency of our cells. And two, there's going to be an increase in uric acid.
Right.
So,
And that you can see that like with a 20-ounce Coke,
yeah,
uh, for sure. So when that happens, uric acid was historically viewed as an antioxidant. But our group, and now it's been confirmed by many other labs, found that when uric acid is generated in the liver, that it actually stimulates a major oxidative enzyme called NADPH oxidase.
hmm,
And that enzyme generates oxidants.
hmm,
Interestingly, the uric acid induces that enzyme to translocate or move into the mitochondria. Our
hmm,
mitochondria are major energy producing cells
yeah.
or organelles. And when NADPH oxidase enters the mitochondria, the oxidative stress knocks out or reduces the activity of various enzymes in both the Krebs cycle as well as in the electron beta fatty acid oxidation.
Got it. So this is a great example of why we need to be careful and not think too simplistically in biology. Uric acid is an antioxidant. It can act as an antioxidant, but at the same time, you're saying it induces this enzyme, which has a pro-oxidant effect.
Right.
Yeah.
And oxidative stress goes way up
Yeah.
in the mitochondria, and you can block that by lowering the uric acid or preventing the uric acid from being formed. And so it is definitely the uric acid that's driving that oxidative stress.
Got it. So fructose ingestion at a sufficiently high level goes to the liver. ATP goes down. There's an ATP depletion effect. Uric acid goes up. That uric acid goes up. It turns on this NADPH enzyme. And that enzyme, just by its nature, it's intrinsically going to promote oxidative stress and reactive oxygen species formation. It also physically goes into the mitochondria. And that's sort of a double whammy, right? Because you've got more ROS production, and then inside the mitochondria, these are organelles that are particularly sensitive to that oxidative stress.
So there's certain enzymes in the mitochondria that are very sensitive to this oxidative stress, and one is known as aconitase in the Krebs cycle. And when you block aconitase, citrate accumulates and activates de novo lipogenesis and fat synthesis. In addition, we see the uric acid inhibits an enzyme called AMP kinase, and this is very important in rescuing ATP levels when they're low. So suddenly you have a mechanism that maintains the ATP depletion by stunning the mitochondria and affecting recovery mechanisms. And so the ATP levels, when they fall acutely, they'll stay low for maybe 45 minutes or an hour before our body recovers. And that period of time is associated with oxidative stress to the mitochondria. And what some of our colleagues have shown is that repeated oxidative stress to the mitochondria damages the mitochondria over time, causes mitochondrial fission and other things. And then you start losing mitochondria. And now you're really affecting the way your body energy balance because the body needs those mitochondria for energy production. At the same time that this happens, the first metabolite of fructose is fructose 1-phosphate. So it gets phosphorylated at the 1 position. And it turns out that this molecule has signaling properties. It's not just involved in energy production, like most metabolites of fructose and glucose, theoretically, right? but rather this thing signals and activates a key enzyme called glucokinase, which stimulates glucose uptake in the liver. And, and so there's this massive uptake of glucose into the liver that's driven by fructose. That um, provides additional fuel. But what's really interesting is that the citrate that is produced by blocking aconitase, which we talked about, as well as um, lactate buildup, turns out to inhibit the classic glycolytic pathway of glucose. And so there's a massive shuttling of the glucose that comes in dramatically into other pathways. And some of those include the pentose phosphate pathway and other pathways. But part of it is driving down uh, an equivalent glycolytic pathway as normal glycolysis, but without any negative feedback. So when you eat glucose, as soon as you start producing ATP, you stop that process. The whole purpose of glucose is to kind of fill up the ATP. And once the ATP levels fill up, this whole process starts to stop. But with fructose, it keeps that glycolytic pathway going, but it's not being converted to energy as much. And
Yep.
so it's shuttled into other
Yeah.
pathways, including fat synthesis and like
Yeah.
lactate production.
Yeah. So if I sort of step back and try to abstract some of what you just said a little bit, when fructose gets into the liver at sufficiently high levels, we talked about the ATP depletion, we talked about the uric acid formation and the problems that causes in terms of oxidative stress and mitochondrial issues. It sounds like a lot of what you're saying is like, if I was to think about this as an engineer, there seems to be an absence of a lot of checks and balances you might expect when fructose is going through here that might be more present on the glucose side. And so we'll get, I think, later into the conversation of why is the biology set up this way, where maybe some of the checks and balances you might want to be there are not actually there, and what that means. But, again, we talked about ATP depletion, uric acid formation, and also oxidative stress. You mentioned that this can induce de novo lipogenesis in the fat synthesis by the liver. What?
Yeah, let me there's kind of a key moment in our research where we had a kind of an explanation for these difference in metabolism that just fits perfectly. And that is challenges the basic concept of what drives obesity. So let's just talk about that. So the classic energy balance theory is that we eat food which provides us calories, which is basically a type of energy, and we use those calories, the carbohydrates and protein and fat we eat, to generate energy, which is ATP.
Mm-hmm.
And then classic idea is that when the ATP fills up to a certain level in the cell, there's a signal to have the excess energy stored.
Right.
And we store that energy as fat or glycogen. And so the idea is that you fill up the tank of gas, and then if there's still food coming in, which is driven by taste and all this stuff, ultra processed junk foods, that extra energy goes to fat.
Yep. And the basic idea here, right? This is the idea of, well, if you want to lose weight, you need to burn more than you consume. And if you consume more than you burn, you're going to save the excess calories for later. The body's set up to burn a certain amount and then save away everything else for a rainy day in the form of fat.
So what actually is shown in animals, which was shown by a guy named Kesey back in the 80s, is that if not counting sugar here, we're not talking about fructose, but if you feed an animal and you can even put a tube into their stomach and force feed them, you can make them fat because you're giving them more energy than they can expend. And this is forced feeding. You can make them fat, but as soon as you pull the tube out, they'll go back to their regular weight.
Hmm.
Likewise, if you fast animals and you make them really skinny, by just not giving them any food, and then you stop the fast, they will go back to their weight. And what's more, they'll go back to the weight they're supposed to be for that time in their life.
Yeah.
So if
Yeah.
it's a hibernating animal, it will go back to increasing its weight in the fall, etc.
Yep. And that
And then,
implies, right, that implies there's feedback, there's regulation, the body can
right.
assess what needs to be
And
done. And
And there are studies in humans. There are multiple studies. They're mainly from the 50s and early 60s, showing the same is true with people. Yes, if you eat too much, you tend to burn it the next day or two. And likewise, if you eat too little, basically we maintain a balance. So, but the energy balance theory says that, oh, the food tastes so good, that's what's driving weight gain. But then why can't we lose weight so easily? Well, it turns out that the fructose story provided the insight for why this theory isn't quite right.
Mhm. Can I ask you a quick question here? So I might
Yeah.
be anticipating something that has been observed. So a key observation here that would throw a wrench in the strict energy balance model would be if fructose on its own can drive the stuff that we were talking about, including de novo lipogenesis, then in theory you might be able to observe with a high fructose diet de novo lipogenesis even in a slight negative energy state.
Yeah, exactly. And actually, we did do a study. So once when we were studying fructose and we found that we gave fructose to animals, that they became big and fat and insulin resistant and diabetic and hypertensive. One of the key questions was, well, maybe the fructose tastes good, so maybe they're just eating more calories and
Yeah.
that's the cause.
Yeah.
So what we did was we pair fed them. And what that means is one group, all animals get the same amount of calories, but one group gets, a sugar-based diet and the other one doesn't. And when we did that, we actually did the experiment with equal calories, but we even did it under caloric restriction. So we put all the animals on a diet. So they're eating 90% of what they normally eat.
And so they should be losing a little bit of weight.
Right. Well, it turns out that when you put an animal on 90%, they just don't gain weight. They don't really lose weight. If
Got
we'd
it.
done 70%, it would have been really interesting, but we just
Okay.
did 90%.
Yeah. So
So
in
it
theory,
stunted because these are growing rats and they
yeah,
should get fatter and bigger, but they stay the same weight.
got it. So they should be holding steady in this condition.
And it turned out that with the group that got sugar had just a minimal change in weight, but it did increase more than the other. And it was not because of increased energy intake, but because the resting energy metabolism falls with fructose.
Got it. So
So,
energy intake was
but it was really mild, it
yeah,
was, but where the difference was in everything else. The rats that were on a diet still became diabetic. They still got super fatty liver.
Hmm.
They still developed hypertension. They still got visceral fat. So even though they're eating the exact same number of calories,
Right.
there was another effect going on that was independent of calories. Now, the weight gain is really driven by calories, but all the metabolic effects like insulin resistance and so forth are not.
Yeah.
So, but now let's get back to the kind of the key explanation for your viewers. So it turns out that the way you induce weight gain is to drop the ATP levels in the cell. When you drop the ATP levels, and you don't want to drop it to crisis levels, So if you drop it to like 30% of normal, the animal goes into a starvation mode and tries to break down everything it can to make energy to survive.
Got it. So that would be like a true
That's not what
starvation
I'm
state.
talking about. That's
Yeah.
not what I'm talking about. So what I'm talking about is you take the ATP levels in the liver and you drop it by 20%. So the animal feels that it doesn't quite have enough energy.
Hmm.
And what's more, it kind of feels like it's at risk. It's at risk of getting into trouble. It
Hmm.
still has enough calories to get by, enough ATP to get by, but it's like gone into a safety mode. It's like the alarm, the light has gone on the panel, gas low.
Right.
You
So the
still
difference
can
would be
drive.
if as opposed to a true starvation state, the mouse's body isn't telling it, let's start breaking down our muscle and stuff to make
Right.
glucose because we're dying. They're saying the body's saying effectively, we're running a little bit low and we might want to go out into the world and just consume more to replenish that now.
Yes, exactly. So what happens is the only way that happens is you have to keep the ATP levels low, which stimulates appetite, and that's been shown in multiple studies. But you also have to block the fat from being broken down to replace the ATP, because normally ATP would be generated by breaking down fat or breaking down glycogen. But what happens is fructose blocks both. It blocks the breaking down of glycogen and the breaking down of fat. And so by doing so, this makes you stay hungry. And so you have to, suddenly you'll eat more than you need. You're actually tricking yourself because your total body energy is high. Your body energy is stored energy plus active energy.
Mm-hmm.
And by dropping the active energy, the sensors in the body are saying we're low. Normally you would break down the fat, but if you block that, basically it tells you you need to start storing fat.
Right. So
And
even though the same number of calories are being consumed, if some of those calories, if enough of those calories are fructose, then the ATP drop acutely in the liver is interpreted by brain and the body as we're running a little low, even though you're not. We should go out and get even more. And
right.
so,
And
this
then
bring
and yeah, exactly. And then as those calories come in, they're shifted to fat through de novo lipogenesis. So fructose blocks fat energy fatty acid breakdown through this mitochondrial oxidative stress, and by blocking fatty acid uptake into the mitochondria. But the main effect is,
Mhm.
ATP levels low, glucose is coming in, and de novo lipogenesis is coming in, and so there's this big push to make glycogen and fat, especially
Mhm.
fat, and it's a brilliant system.
Yeah.
And so animals like to use this system to prepare for times when there's starvation, like before hibernation or
Right. When you want to put on as much extra energy as possible in the form of fat.
right, right.
Yeah.
So
So,
that's why, I mean,
yeah.
bears, for example, will eat lots of fruit and honey. They'll seek it out because it helps trigger them to eat more. And
Mhm.
part of that is, you know, through this fallen ATP activating this process. So one of the things we discovered too was that there's a liver-brain connection. And when fructose is metabolized in the liver, the brain will suddenly become resistant to leptin. And leptin is the main satiety hormone.
Hmm.
So when we eat, leptin levels go up in the blood and they activate an area in the hypothalamus that controls food intake. And what happens is when you give fructose to an animal, after about 2 months, they become leptin resistant. And then not only are they eating because the ATP levels are low, but once they're leptin resistant, they will just try to eat as much, a lot
Yeah.
of food.
Yeah. So it's a
It,
double whammy.
double
Hunger
whammy.
is stimulated by the drop in ATP, but then
Correct.
over time, the satiety signal that would come from eating
Right.
more food is it broken, and so now you're kind of in a vicious circle.
So,
So,
yeah,
so
go
I want to
ahead.
think a little bit more about why the system is set up this way, and we've already started to talk about it. So, from an evolutionary standpoint, you've mentioned that this is actually a great system if you're an animal that lives in a seasonal environment, and there's times of food scarcity where you need to put on a lot of weight quickly to prepare for that upcoming scarcity. Can you say a little bit more about what you've called, I think, the fructose survival hypothesis, and why this
Yes.
whole system is set up this way in animals?
Yes. So when an animal hibernates, before they hibernate, they'll completely be regulating their weight very well during the summer, during the spring and summer. And then in the fall, before before they hibernate, they'll suddenly gain weight. And this will be a massive weight gain associated with leptin resistance, which is what we see in people with obesity. They get fat, they eat more, they get fatter, but they're also getting fatty liver, they're getting insulin resistant, and they're getting all the features of metabolic syndrome. And we were taught, at least I was taught, that metabolic syndrome was a disease. It's kind of like a condition that leads to high blood pressure and diabetes, and it does. And so it's sort of considered pathophysiologic. If you have metabolic syndrome, you're at high risk for developing obesity, and it's not a healthy thing to have metabolic syndrome. But these animals are purposely developing metabolic syndrome. So the question is, why is it, what is it about it that could be good? And it turns out that when there's no food around, like what happens during hibernation, is the animals won't have access to food or water for months. They won't eat, they won't drink, and they will not, and they will just hibernate. They go into torpor and they live off their fat. And it turns out that when you burn fat, you not only produce calories, but you produce water. And when you burn fat, you make a thing called metabolic water. There's no water in fat, but when you burn fat, you produce water. And I've done a lot of work in this area, and it turns out that animals that are hibernating use the fat to provide them the water they need, as well as the calories. So when you're storing fat, you're actually storing water in a sort of sense.
Hmm.
There's not actually water there, but it's there to protect you, just like the camel that has the hump of fat that it can use as a water source, or a lot of desert animals have fat on their tail to provide calories and water when they need it. Okay. Whales have lots of fat. They don't drink water, so they get most of their water from their food or from the fat they burn. About a third of the water they get is from the fat they burn. So fat is a source of energy and water. And so if you're going to be in a period of time where there's no food around, you want to accumulate fat because it's going to provide those two key things.
Mm-hmm.
Well, what about insulin resistance?
Yeah, yeah.
Well, the brain's preferential fuel is glucose, preferentially. We know that it can survive on other things. Maybe some of the other things have some advantages, but the brain prefers glucose. And if you're starving, you can get hypoglycemic. You can get your blood glucose levels fall because you deplete your glycogen stores. And so you can get hypoglycemic. And if you're trying to survive as a starving animal, you don't want to be hypoglycemic because you need your brain to function adequately as long as possible. In fact, the brain is more important than the muscle because without a brain, you're gonna die with the first predator. So what insulin resistance does is it reduces glucose uptake in the muscle significantly. So that's the muscles become resistant to insulin. And when that happens, the muscles don't get as much fuel. You actually decrease your energy expenditure because the insulin resistance is reducing the amount of glucose that it's using.
Yeah.
But there's another benefit. So not only does it help conserve energy, when the glucose goes up in the blood, it tends to stay in the upper end of normal and slightly elevated. That provides a fuel for the brain because much of the brain doesn't use insulin. Some of the brain does use insulin, but the parts of the brain that don't use insulin can benefit with that insulin resistance. Now, it turns out that the part of the brain that likes glucose and is not insulin resistant is a part of the brain that's involved in foraging and in seeing food. So, for example, like the ocular cortex, you want to be able to find food when you're starving or when you're preparing for hibernation. So it turns out that fructose specifically activates foraging pathways, and that's the insulin independent pathways in the brain. But when you induce insulin resistance, the areas of the brain that require insulin turn out to be those involved with recent memory and the cortex, where it's like being able to think things through things. And so what happens is when you eat fructose, you actually stimulate foraging so you can find food, but you're reducing the energy to the cortex for thinking through, is it dangerous or not? Your recent memory is impaired because they don't want you to remember that there are predators there. You've got to be bold and impulsive. You got to be able to look in all around and make quick decisions. And that's what fructose does. So if you give fructose, cortical blood flow drops. You give glucose, cortical blood flow goes up. If you give glucose and you inject it into the brain, ATP levels go up in the mouse, and the mouse quits eating because its satiety signals activated. You inject fructose in the brain, ATP levels fall, and the animal starts looking for food.
Got it. So the same drop in ATP that we talk about in the liver can happen elsewhere in the brain as well in response to fructose.
Absolutely.
And that's used as a signal by our cells.
Right. So basically, these two sugars are so critical to biology. We have focused on glucose because it's the main sugar in our blood, and insulin diabetes is one of the main diseases we have. We know everything about glucose, but fructose metabolism has been kind of ignored in the human. But what fructose does is it stimulates processes associated with trying to store fat. So if you're an animal and you want to store fat, what do you want to do? You want to increase, you got to be hungry. You've got to be foraging, looking for food. You've got to be able to ingest as much food as possible, like leptin resistance. My friend Marcus Goncalves shows that fructose actually increases the villi area in the intestine, so you absorb more food for the same amount ingested. I mean, it just goes on and on. The list goes on and on. And fructose is the fuel that does that. Fructose is the one that stimulates de novo lipogenesis. It's the one that blocks fatty acid oxidation. It does all those things. Now, glucose can do that, but much less, much less.
Yeah.
And when it does it, it's likely because the glucose is being converted to fructose.
Yeah. So basically, one way of repeating some of this is instead of just thinking about metabolic syndrome, obesity, insulin resistance as merely something being broken and something that's wrong, you can, if you look at this sort of through an ecological evolutionary lens, these are actually features. They can be,
Yes.
these are adaptations that animals in the wild have to prepare them for the conditions they have to survive through, such as a long winter where food and water are very scarce. And
Right.
so the modern environment for humans, it's almost like we're in this kind of perpetual winter, as I think some people have called it. We're seeing all of these signals of abundance, including fructose, and our body's doing what the bear's body is doing in some sense. It's
Yep.
packing on all of this extra fat
That's
because
right.
it's getting signals that signal future scarcity, of course, that never comes.
Right. And here's the key. And this is why I wrote my book, Nature Wants Us to Be Fat, is this is a survival pathway, as you just pointed out. So, but what happens when you stimulate foraging chronically? You are inhibiting blood flow to the brain, you're inhibiting blood flow to the hippocampus, and you're inducing insulin resistance in these sites. And you're inducing mitochondrial oxidative stress because fructose is being metabolized in the brain.
Mm-hmm.
So you get mitochondrial, you get oxidative stress, you get inflammation. And guess what? You give chronic fructose to an animal, they develop Alzheimer's. They get amyloid plaques, tau protein aggregates, memory problems, all the things that you see with Alzheimer's.
Yeah,
And so
yeah,
it turns out that the part of the brain that's insulin dependent, which is where the insulin resistance is induced by fructose and where the ATP levels fall first, those are the first sites where Alzheimer's manifests.
well, I
And
want
the
to,
wandering response in Alzheimer's is probably a foraging response.
yeah. So,
And so you can suddenly the foraging response becomes behavioral disorders, ADHD, bipolar disease, all these things can be seen as overactivation of this pathway.
yeah.
And dementia is the ultimate bad outcome.
Mhm.
You
So
look at, yeah,
if we go back to thinking about, animals like bears that have to hibernate, a big difference between bears in the wild and humans today is that fruit is only seasonally available. So there's a natural
right.
environmental limit that the bear can't get around. The bear can't go to the supermarket whenever it wants to and get access to all that fructose all year round. Two, the fructose is only available in a physical fibrous food matrix, which is going to limit its absorption and metabolism in ways that a lot of the foods we're exposed to don't. Can you say a little bit more about the importance of how the natural environment limits fructose, both like seasonally and packages it up in physical food matrices that have fiber in them, and how the modern foods that we consume that don't have those things are tripping us up in certain ways?
Yeah. So animals have gotten wise, at how they do this, and so has nature. So trees, fruit trees will the fruit tends to occur in the fall. And as the fall progresses, the fruit matures and the fructose content goes up. So fruit tends to get sweeter as it matures. A unripe banana is not particularly sweet, but as it matures, it ripens, the sugar content goes up. And a lot of other things that block the effects of fructose, like vitamin C actually blocks some of the effects of fructose, and flavonols and epicatechin, all these things that are in fruits, but they tend to go down as a fruit ripens. So the plant doesn't want the animals to eat their fruit until the fruit's matured and the seeds are good, because then when the animal eats it, the seeds end up in the poop and help the tree. So it kind of times the fruit to ripen as fall progresses, which is exactly what the animals want. And they won't eat small amounts of fruit like we do. They'll eat as much as they can so that they get that fructose load. And they'll wait till the fruit's really ripe when the sugar content really goes up. And we don't really like it when it's mushy and that kind of thing. So what happens is nature has this timed. There's hummingbirds have these long bills, so they can actually pull out nectar, which is basically sugar water. And they can get fat in a day. They eat mainly sugar, and they get the fattest liver of all birds, and they get they become diabetic during the day. But during the night, when they don't go out for they can't. I don't know, but they don't eat at night.
Yeah,
Then
yeah.
they burn off all the fat because they have such high metabolism.
Hmm.
And in the morning they can go again, and their blood sugars will drop. And if they drops too much, they'll go into torpor. They go into kind of a sort of hibernation for 3 or 4 hours. They'll just sit on a branch.
Wow.
So that nature has got it all worked out. But we learned how to make refined sugar and high fructose corn syrup. And because we have a taste for it and we have a craving for it, we mix it into all these foods and we mix it into processed foods and we coat it on sugary steaks and I mean it's added to
Yeah,
sauces and it's everywhere. And so food is available 24/7. You can go down to a grocery store, you can eat an ice cream at 3 a.m.,
yeah.
and
So
and so we're just
we became
yeah.
clever enough to do all of these things, but we weren't clever enough to realize what the consequences of our cleverness would be.
Yes. Yes, we're victims of our own. And we also discovered that humans are particularly vulnerable to sugar. We're much more sensitive to sugar than the mouse, for example. And this was because we had a mutation that occurred in uric acid
Yeah,
like 12 million years ago during the period of starvation. And this mutation probably helped us survive.
yeah,
It was during a time of seasonal starvation, so that there was time to during the much of the year to store fat
yeah.
if we could, and then
So I've actually written about this. Basically, what you're saying is, long, long time ago in the ape lineage, there was a starvation period, let's say. We had a mutation in the uricase enzyme, which basically means we're going to make more uric acid or break it down less than other creatures. That probably helped us store fat and survive that period. But
for
now,
sure,
today, that feature of our metabolism is probably hurting us.
yes, absolutely, yeah, yeah. We I worked with a paleoanthropologist and a wonderful human being, Peter Andrews, who is probably the world expert on this period of time. Actually, was able to show that these ancestors of us were going through seasonal starvation because the fruits were not strong enough to. But at one point, there was fruit available all year round, but then it became less available during the cooler months, and so the animals started starving, and he actually confirmed that there was starvation going on in our ancestors and certain regions of the world where our ancestors were actually they actually went extinct. But a mutation occurred around that time that allowed us to process fructose more effectively. And we showed that we resurrected the extinct gene, and we did a lot of studies to prove this. But yeah,
So we talked about this fructose survival hypothesis and the fact that the way fructose is metabolized can stimulate animals to go out and forage more, to get more calories, to store as fat to save up for later. We talked about the ATP depletion, the uric acid formation, the unique sort of sensitivity we have and other apes have on that side due to our evolutionary history, and the oxidative stress burden that fructose metabolism can place on our mitochondria, which can break them over time if we're eating a lot of fructose all the time. One thing that's interesting to me here about all of this fructose stuff is it kind of lines up in an unfortunate way with something I know about one of the other big ingredients in the modern food environment, which is the omega-6 polyunsaturated fats. Those fats can be obesogenic and promote fatty liver. They can promote food intake via endogenous cannabinoid signaling, and they can render mitochondria more sensitive to oxidative stress, which, as we discussed, can go up in response to fructose intake. And it seems to be, probably not a coincidence that two of the major features of the modern industrial food environment are high omega-6 content and high fructose content. Do you think that the combination of high omega-6s and fructose is particularly problematic?
yes, I do. Let me talk a little bit about this because some of the work we've done on this we haven't published, but well, we've published it, but we haven't uh, discussed it in this light. Um, so it turns out that when animals are hibernating, before they hibernate and they're gaining fat, we believe that the fructose is the driver, but when they start becoming leptin resistance, which is a fructose effect, leptin resistance is associated with increased craving for fats, especially polyunsaturated fats like omega-6. And so there is this kind of stimulation of fat intake that goes along. Now, the reason that has a survival benefit is, you're trying to put on fat. If you're on a keto diet and you're eating a high-fat diet, you don't gain weight because you're controlling your appetite. But if you have your appetite affected by fructose so that you're hungry, then when you're exposed to a high-fat diet, you will eat more fat and you'll get more calories because you're not regulating your weight. So in other words, if you take animals and you give them fructose and make them leptin resistant, and then you give them fat, they get really, really fat very quickly. You can even stop the fructose and they'll get fat because the leptin resistance carries over for a few weeks. But if they are not leptin resistant and you give them lard or something like that, they don't gain weight. So it turns out that, a big part of gaining weight with fat is to have leptin resistance present from fructose. Now, when you give fructose to animals, or if you, particularly when we did these studies, were mainly with raising uric acid, it turns out that you start activating enzymes in the prostaglandin pathways that are associated with inflammation.
And prostaglandins are inflammatory lipids that come from omega-6s.
1 second. Can we stop for a second?
Yeah, go for it.
Yeah, my dogs are barking.
So I want to point people to some articles I wrote while we're waiting here. I have an article about seed oils and fatty liver disease, which I think ties into this very well. So just think about some of the synergies we're talking about here. Fructose can stimulate hunger pathways and oxidative stress. Lipid peroxidation comes from, lighting those PUFAs on fire. And there's a lot of dots that can be connected here, as I think we're going to discuss. But the modern food environment is characterized by high omega-6 and high fructose content. Are you back, Rick?
Yeah, I'm back. Sorry.
Okay, just kind of talking.
So anyway, so it turns out that when you activate this fructose uric acid pathway, you steer the polyunsaturated fats into inflammatory prostanoids like thromboxanes. And those are very inflammatory. So what I believe is that there are these studies with omega-6 where they'll give it to young, healthy people and they don't really see much negative effects. And so there's this literature where people are saying, omega-6 aren't bad. But if you give omega-6 in the setting where fructose and uric acid are involved, the omega-6 are precursors for the prostaglandins. And the enzymes, there are good prostaglandins and bad prostaglandins, but fructose and uric acid activate the bad enzymes. So when you take omega-6 with fructose and uric acid, it's going to be pro-inflammatory. But if you gave omega-6 to people who are very healthy, you may not be able to see that because the omega-6 is just the precursor.
Yeah. It's just one of many great examples of why you have to be able to think about biology and metabolism in a context-specific way. Right. The combination of all these things can be really critical.
The other thing that's really interesting is, the omega-3s. There's really nice data showing that they kind of counter some of the fructose effects in animal models. I still don't really know the mechanism, but it might be through the fact that omega-3 lead to healthy prostanoid type compounds.
Well, I just had Floyd Chilton on and we discussed this. Among other things, it's because the omega-3s and 6s compete for processing from the same enzymes, we know that if you boost omega-3 consumption, tissue levels of omega-6s go down and vice versa.
Yeah.
So, these things do have this kind
Yeah.
of balancing act that happens, and it starts to make a lot of interesting sense of a lot
Yeah.
of biology.
Yeah. One of the great things about this hypothesis of our group, this fructose survival pathway, is I can look at a seed oil paper and I can explain a negative versus a positive
Yeah.
result.
Yeah.
And also I can explain how the energy balance theory is important, but how it, kind of misses some of these things. And I can discuss Gary Taub's stuff and show
Right.
how it,
Right.
it
Yeah,
interplays.
nobody's saying that the laws of physics need to be violated and total
Right.
calories don't matter, but the identity of those total calories also
Right.
additionally matters in other ways.
Yes, exactly. If your weight's regulated, if you can regulate your weight, that takes into account, and so you'll get full before you really gain that extra weight.
Yeah, yeah,
Yeah, it's kind of fun to think about biology in evolutionary terms, and also in to walk in, and when you hear someone's got a very strong hypothesis, listen to it, and there's usually some very good points that they make. And then the question is, but how does that interplay with in an evolutionary basis, and how does it interplay with what we've discovered? And it's fantastic how they all kind of fit into pieces. I wrote a paper um, just this last year, or maybe 2 years ago, where I took all the main obesity hypotheses and I put how they work on one piece of paper, or one figure, and showed how they all interact, and how the fructose survival pathway actually can explain the protein sparing and the energy balance and the carb insulin models. And I think everyone is partly right. And probably I'm only partly right too.
yeah.
But
Well, it's like that. It's like that analogy here sometimes where I forget what you call it, but everyone's touching a different part of the elephant, and they don't
right,
realize it's an elephant yet. Someone's touching the
right.
trunk, and someone's looking at the ears, and yeah,
Exactly.
yeah,
That is how it is. I'm absolutely convinced. And I think there's only one big elephant, but there could be more than one big elephant. But right now, I think the big elephant is this fact that carbohydrate metabolism has two pathways. A pathway that's for kind of immediate energy, which is the glucose model, and one for stored energy, which is the fructose model. And interestingly, here's a fantastic thing. If you raise blood glucose, like in a person with diabetes, or if you give a high glycemic meal, like if I eat pancakes, and I have a glucose monitor, so I can actually look at my own glucose and see it go up. I don't like it going up, because when it gets over about 140, I know much of it's being converted to fructose, including in the brain, because the high blood glucose remember, it circulates everywhere. And fructose doesn't quite get there, but high glucose does. And so it turns out that these rise in blood glucose is the best way to increase fructose levels in the brain. That the brain does not do well with high glucose. And there's a guy at Yale who showed that in humans that if you clamp blood glucose levels to elevated levels, the brain starts making a ton of fructose. And fructose levels are high in the brains of people with dementia, and high in the brains of people with bipolar disease and so
yeah,
forth. So I
yeah.
think, yeah, I think that this is another, but again, it's a survival mechanism. If you become insulin resistant, not only is that a survival mechanism, but the high glucose allows for more fructose production. So it's an amplifier
Yeah. Did
of
you
the survival system.
did you happen to see this paper that came out recently about inulin and fructose in rodents and the microbiome?
Yes.
Yeah, that was
So
I love that story because I was reading it side by side with the review you just came out with, and it tied some things together in a really nice way, in my opinion, which was that, fructose from fruit is bound up with inulin and fiber. Part of what that does is limit the absorption rate, but also part of what it tended actually happened to do was stimulate the microbiome in a way that, long story short, it actually stimulated the glutathione pathway in the body to help buffer the body against the oxidative stress induced by fructose and prevent lipid peroxidation in the liver. So it was another example to me of how some of these things tie together: fructose biology, lipid biology, and
There is this
fiber.
there is this mix though. So inulin is called a fructan because it's like a series of fructose molecules, linear. And they actually do, at least in that study, it looked like it was protective because it basically stimulated fructose metabolism to get rid of fructose in the gut.
By the microbes.
Right. But there is a problem in horses, and we've done some studies in horses. And one of the problems with horses is that they can get fat
Hmm.
and insulin resistant, and they can get this disease called laminitis, where they basically can't walk. And it's a real problem among thoroughbreds. And it was shown that what happens is um, in the summer and late spring, some grasses will start to accumulate fructans, probably as a survival mechanism for the plant, but that's another story.
Hmm.
And these fructans are mainly leavens, so they're not inulin, they're a different type.
Mm-hmm.
But if you feed those fructans to animals, to horses, they will develop metabolic syndrome and laminitis. And it turns out that mammals can't break down fructans, but some enzymes in microbes can.
Hmm.
And it's been discovered that there's a strep species in horses that breaks down the fructans to fructose and can induce, likely has a role in inducing metabolic syndrome and laminitis. And I've written a little bit about this, but so the trouble is inulin is a fructan too, but it's not the one that's in those rich grasses, the bluegrass of Kentucky. And so not all fructans are good.
Yeah, yeah.
So inulin may be good, but we also don't know if maybe at high doses, so the horse will eat huge doses,
Right,
right?
right, right.
So the question is, um, in the study that was done by my friend, I mean, I know the group. Um, the question is, there probably is a dose of inulin that stimulates fructose metabolism and is
Right.
good, right?
Yeah.
But if you give too much, there's going to be an overflow of fructose and it's going to get absorbed, and then that might be a problem. So we have to be very careful.
Right.
I don't want people running out and, eating
Scooping
inulin.
grams of inulin into every drinking.
Yeah, yeah.
Yeah, yeah.
It's,
Yeah, I mean, well, I think a key principle for me is, the absolute dose and the relative balance of things almost always matters. You can't think about these things in isolation. You got
there
to
you
think
go.
about them, in context.
There you go.
I want to spend the rest of the time we have talking about brain stuff because I think this is important, and I know that you've got a lot to contribute here. Say a little bit about endogenous fructose to get us to the brain. Remind people, where does this endogenous fructose come from in our bodies?
Yeah. So there's only one enzyme in humans that makes fructose, or one pathway, and we call it the polyol pathway. And basically, the only way to make fructose is to make it from glucose. So it all starts with glucose. And then the glucose can be converted to sorbitol, and then the sorbitol gets converted to fructose. And those enzymes, the enzyme that converts the fructose to sorbitol, glucose to sorbitol, is sort of the key enzyme. And that enzyme normally is minimally expressed in our body, except in certain areas of the body. But when you eat a lot of sugar, you induce that enzyme. And once you induce that enzyme, then it can convert glucose to fructose pretty readily. Now, the things that can convert it, that stimulate that enzyme, include Western diets, high glucose foods can do it. So diabetics who have high glucose in their blood, they have high levels of this enzyme. So they're making a lot of fructose. Likewise, salty foods will activate this enzyme. Alcohol activates this enzyme. We recently showed that the fatty liver from alcohol is actually from fructose. The alcohol doesn't make fructose. The alcohol activates the enzyme that converts glucose to fructose.
Mm-hmm.
So when you're drinking, it's those pretzels that you're eating with the alcohol that are being converted directly into fructose because it's salty glucose. Salt and glucose are being ingested, and alcohol. So it turns out that you can activate this pathway even with stress. It can be activated with trauma, with ischemia, or like a heart attack will induce this pathway in the heart. Acute kidney injury is associated with activation of this pathway. It turns out that whenever there's any kind of anything endangering you, this pathway gets activated, which sort of has a survival sense. And salt, the reason salt does it probably is because salt increases our osmolarity and makes us dehydrated. We actually aren't losing water, but we're increasing the salt concentration in our blood to make us want to take in water.
Right. Okay.
And so that makes us appear to be dehydrated. And the animal will want to store fat in response to that because that's another source of water. So not only does it want you to drink water, it wants to store the water. And the way it does that is by making fat. So fat is associated with salt as well as sugar. And when we looked at the data in the literature, we were surprised because we found that salt intake is strongly linked with obesity, and ultra-processed foods mix salt and sugar together. French fries mix salt and carbs together. So you've got the salt and the oil, the fat and the carbs. The glucose and the carbs gets converted to fructose. It's like the perfect food because raises the glucose rapidly, converts to fructose, makes you, like the fat all the more. The salt stimulates. So that's why there's a big difference between salted potatoes and non-salted potatoes. And so it turns out that a lot of the flavors we like, the taste we like salt, is also probably has an evolutionary basis to help us identify foods that may help us store fat in the days when there was not a lot of food around. Likewise, umami is another taste. Umami is this savory taste. We have taste buds for it. But it turns out that the foods that are savory are almost all foods that basically they're the foods that raise uric acid. It's like amazing. I looked at umami and I looked at foods associated with, that raise uric acid, and they're the same. It's the same group. So I think that and it turns out that when you raise uric acid, you activate this fructose pathway, the aldose, the polyol pathway. So unfortunately, when I started reading this, I go, oh my God, and putting it together and doing studies, we kind of realized that this survival pathway is a very central pathway that animals have evolved towards. So to find foods that can help you store fat is something that animals like. And then because we have taste buds for those, we end up not unwittingly eating foods that can activate this pathway. And the big activator for sure is sugar. High fructose corn syrup, that's by far the biggest one.
Yeah.
But
Do you
chronically salt and umami probably can do it too.
Yeah. So sometimes, I've had discussions with people, and we're talking about sugar, we're talking about fructose, and we talk about high fructose corn syrup and things like that. Say, well, it's 55-45. Sucrose is 50-50. That's not a big deal. Do you Do you think
We
that
we did a study in humans where we gave soft drinks that were exactly identical, except one was high fructose corn syrup and one uh, was table sugar. We did that study, and even though there's a 5% difference, it was enough to show a difference in blood pressure response and uric acid response. But there, we can lose the forest for the trees.
right, right.
And both of these, sugar and high fructose corn syrup, they're both really, really bad when you eat a lot of them.
Mhm.
And it's absolutely true. High fructose corn syrup is worse. We actually did another study where we did this and we gave high fructose corn syrup with 50-50 glucose fructose versus sucrose, and we pair fed animals, and we got worse fatty liver with the high fructose corn syrup.
Really? So the only difference
Even
there would have been the monomers versus the dimer of
yeah,
sugar.
and there is a difference because
Wow.
a dimer will have half the osmolarity of two monomers.
I see. Okay, I didn't know
And we
that.
know that the osmolality activates this amplifying pathway, the
Hmm.
aldose reductase pathway.
Yeah, yeah.
So probably that explains it. But I don't want people to think of high fructose corn syrup as being the only evil one in the
Right,
room.
right.
And it is true. One way you can kind of look at this is to compare how long it takes to become fat. And sugar and high fructose corn syrup are right up there. They get you fat very quickly, but you can also get very, very fat with just increasing the salt, but it takes like 3 times the time. Instead of 2 months, it takes like 5 months. And umami is actually quite potent, but we don't eat enough umami. So if you, on a gram per gram basis, umami is probably worse than sugar, but we eat 1/10, 1/15 of umami compared to sugar.
Mhm.
So umami is not a big player unless you like in certain regions of the world, like China maybe, where umami is probably more important.
Got it. In the time that we have left, do you want to discuss at all any of the stuff that you're working on or connected to related to dementia and fructose? We've talked about it a little bit, but do we want to dig into this a bit more for people?
Yeah, we could go into this a little bit. So um, the story of what causes Alzheimer's has been extremely critical important topic because this is becoming a more and more common disease. It was thought to be rare back in the 1950s, and now it's like a major cause of death, and it's probably one of the worst diseases to have, not only because of what happens to you, but for those who love you, because there's nothing like seeing you lose your cognitive function. So, the focus was on the amyloid plaques because this was kind of the key finding that kind of separated it from other dementias. And also there are these things called tau protein aggregates that could be found. And so initially the pharmacology industry, pharmaceutical industry, I should say, focused on trying to find therapies that could reduce the amyloid plaque or block the formation of tau protein aggregates. And I think at one point there were 26 or more compounds being tested, and the vast majority failed to show a benefit. Some of them show very mild benefits, but nothing to write home about.
Mhm.
And so the realization was that the amyloid deposition is probably a late manifestation of dementia, and we should look at earlier stages to see what actually is going on before the amyloid plaques become so prevalent. And three major findings were made. One was that the brain becomes insulin resistant. And as mentioned, the certain areas of the brain are sensitive to insulin. Some of the areas are not, but the areas that are normally sensitive to insulin become resistant to insulin. The second issue was that there were mitochondrial dysfunction and mitochondrial oxidative stress that was seen. And the third was that there was the discovery of inflammation in the brain, what we call neuroinflammation, and activation of the macrophage of the brain, which is called the microglia. So these three became the focus of the next range of attacks, and people are looking at giving drugs to block inflammation or to improve mitochondrial function or to try to find ways to improve the insulin resistance. And even things like intranasal insulin was being given in Seattle, where you are. And so there are all these kinds of approaches. But I was studying fructose, and I found that, systemically, the key feature of fructose is insulin resistance. That is what drives insulin resistance systemically. That is the main nutrient. And we show that in so many different ways. And it causes mitochondrial oxidative stress. In fact, that's key to how it works. And it causes inflammation. So I realized that there was a parallel. So when we started looking at people with Alzheimer's, and other groups did most of this epidemiologic work, it was not surprising to see that the foods that are associated with fructose, like sugar, high fructose corn syrup, sweets, high carb diets, were also linked with an increased risk for Alzheimer's. And not only that, but even things like high salty foods were linked, and all these kind of things that we were identifying, alcohol. And so the next step was to see what happens with animals when they're fed fructose. And if you give fructose or sugar to an animal, they have trouble getting through a maze. Or if they're diabetic, for example, their glucose levels are high, which we know can generate fructose, that you can show cognitive dysfunction. They can't learn. Normally, a mouse, if you put it in a maze, the first time it can't find its way very well, but they learn each time how to get there a little bit quicker. But not if you feed them high sugar. And not only that, but my friend Gomez Panilla did some studies where he showed that if you give sugar to animals, that they develop mitochondrial dysfunction, insulin resistance, and neuroinflammation. And then a group in Egypt showed that if you give the fructose for 18 months, they actually get neurofibrillary tangles and tau protein aggregates and amyloid in their brain in addition to cognitive dysfunction.
Mm-hmm.
So pretty strong evidence. And then a group recently showed that if you had a diabetic animal with cognitive dysfunction, if you just injected an inhibitor of fructose metabolism into the brain, you could improve the cognitive dysfunction. And so that all that was pretty strong evidence that this pathway is involved. So what about humans? So in humans, it turns out that you can find high fructose levels in people with Alzheimer's. It's like six-fold higher than
Wow.
in age-matched controls, along with elevations in sorbitol, which
Mm.
is another metabolite, as well as activation of AMP deaminase, which is the key enzyme. And uric acid is found to be linked with Alzheimer's by Mendelian genetic studies, and was found to be the only key metabolite that really predicted Alzheimer's in two studies. And so I think that we've got a pretty strong case that this is it. And then there's that evolutionary thing, which was really cool. So with David Perlmutter, Dale Predison, and others, we started thinking about this and realized that the areas of the brain that are affected by fructose are also the areas of the brain that are the first to go with Alzheimer's, and also are the areas of the brain where inhibiting blood flow and causing insulin resistance stimulates foraging.
Mhm.
So we
Remind us what
realize
those
that
brain
it's all
regions are.
it's part of this big foraging response. And then there's this thing where 7 out of 10 people with Alzheimer's kind of wander off, and that seems to be like a foraging response.
Wow, I never,
Very,
yeah, that's interesting.
yeah.
Yeah.
So I think, when you look at hypotheses for Alzheimer's, I don't think there's anyone who's ever had a hypothesis that explains everything from the beginning to the end. But we have a hypothesis that explains what the epidemiology is, why the diets do it, how it works, animal data,
And the
human
behavioral
evidence.
symptoms and manifestation of
Yeah, and go
disease.
from A to Z. We can go from A to Z. We can
Hmm.
explain the inflammation. We can, so I think, and we even have an evolutionary mechanism.
Right, right, right.
And so I think I really think this is the cause. And I do believe that and now we've discovered that the key cell in the brain that's involved in this is the microglia. And it's got that special transporter for fructose, and
Yep.
it gets activated by fructose. And, so I'm very excited because I think fructokinase inhibitors that get into the brain will be the first major effective therapy to prevent Alzheimer's. I believe that. That is a hypothesis. So for those of you who are listening, know that but it's based upon this data.
Mhm. In the time we have left, so everything that we talked about with regard to fructose, very interesting stuff. We covered a lot of ground. Let's now talk about the reality that it's 2026. We all live in the towns and cities of the modern age. There's a lot of fructose around. How would you start to advise someone around managing their fructose intake in a realistic fashion? What would you say around added
So
sugars
here,
versus whole foods versus timing and seasonality and things like that?
so here's the first thing. The first thing is that uh, you know there's obviously foods that are high in sugar, especially soft drinks. And we found that if you drink sugar, you get a bigger load to the liver and a higher concentration, so you get a greater ATP depletion, and it activates this process much more rapidly. So cut out liquid sugar. Cut out liquid sugar. Reduce your alcohol intake because that can be converted to fructose. And try to cut back on sugar and high fructose corn syrup. Number one. Number two, all fructose comes from carbs, including endogenous fructose, or fructose you make is coming from glucose. And the main way that happens is by eating high glycemic carbs. So things like pancakes, rice, potatoes, bread, especially if they're salty, salted pretzels, cut those way back. And in fact, if you are on a keto diet, you are lucky because that will block a keto diet will block glucose and fructose pathways. So you basically block the survival pathway. You block that pathway by just going on a keto diet. But can you maintain a keto diet? Many people can't. Can I? I cannot.
Yeah, but you
But
but you might not need to
cutting back
maintain it forever.
carbs.
Yeah.
Yeah. And maybe one day we'll have a fructokinase inhibitor. I think the GLP-1s are working on this pathway too. It's for another discussion, but yeah, there's a hormone that's involved in the fructose pathway called vasopressin,
Mhm.
and GLP-1s suppress that. But that's really another conversation. So
Well, I think we've already covered a lot of ground. Rick, I want to thank you for your time. This was fascinating. This ties into a lot of podcasts I've done in the past and a lot of my writing on the Substack. So if people want to check that out, I will put links to some of those synergistic episodes that I've done on here. And then, yes, Rick, one more time if you want to talk about your book.
yeah, I do have a book called Nature Wants Us to Be Fat. It talks about the survival hypothesis. It presents the data for it. It tells the story of its discovery and how the science and the series of kind of discoveries we had when we were going through this, and it provides dietary recommendations. And yeah, nature wants us to be fat.
All right. Well, Professor Rick Johnson, thank you very much for your time.
Thank you, Nick. Bye.
Hey everyone, a great great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.